Alright, so it's been a long while since I've made one of these, so this one's about protein targeting, which is basically what happens to proteins, where do they end up, how do they end up where they are. If we start off with a protein in the cytosol, there are two possible routes it can take to end up wherever it's going. We have the post-translational transport, which is basically transport that happens after the protein has been translated in the ribosome. And there are 4 places it can go from the cytosol. The protein can either go to the mitochondria, can go to the peroxisome, it can go to the chloroplast, if it's a plant cell, by the way, or it can go to the nucleus. Now, the alternative is co-translational transport. Right, and this is transport that happens literally as the protein is being translated. And this occurs via what's known as a signal peptide. So we have our protein starting off in the cytosol and it goes to the endoplasmic reticulum. And from the endoplasmic reticulum, it is sent into the Golgi. And this is through vesicle transport, so we use transport vesicles. Okay. From the Golgi, it has a number of places it can go. It can be excreted, so it can be exocytosed, it can be excreted. Plasma membrane, so it can be part of the membrane that can embedded within. I'm actually running out of space, I'm going to write this elsewhere. And can be sent to the lysosome or to the other parts of the endomembrane system. So first of all, we're going to talk about all of these in a lot of detail, but first we're going to start off with the co-translational side. So we're going to focus on this first of all. So proteins are fed into the ER during translation if they have an amino acid sequence known as a signal peptide. Okay, so normally the signal peptide that sends a protein into the ER during translation is a series of hydrophobic amino acids, usually found at the end terminus of the protein. Doesn't have to be, but it's usually found there in co-translational transport. So I'm going to draw a diagram that should hopefully explain this in a bit more detail. Okay, so this may look a little complicated, so we're going to do it step by step. This right here, this big thing, is the ribosome. That is the mRNA. You can see the 3' end and the 5' end, or the 3' and 5'. This right here is a signal recognition peptide, okay, the SRP. What it does is it binds to the signal polypeptide as it comes out of the ribosome. So you can see this polypeptide coming out of the ribosome, and at the final end we'll have the signal polypeptide, like that would be the final part. And the SRP binds to the signal polypeptide as it comes out of the ribosome. The SRP brings the ribosome to the ER by binding to a receptor on the ER surface. And this receptor is associated with other proteins that make this pore, which is known as a translocon. The ribosome will then continue translating and feeding the polypeptide through the pore and into the ER lumen. Okay, and an enzyme which is associated with the pore, known as signal peptidase, will go and snip off the signal peptide, and your SRP will be displaced and recycled. So that's what happens. Okay, now that's how we get stuff from the cytosol into the ER. Some membrane proteins can actually remain in the ER So if you're a protein that wants to remain in the membrane of the rough endoplasmic reticulum, you need to have what is known as a stop transfer sequence, and this is usually a sequence of 20 to 22 amino acids, and they will basically make the protein remain as transmembrane segments, so they will just remain in the ER membrane. But most will go into the lumen of the ER, and they will be most of the time glycosylated. Protein folding will take place. Now, what happens to proteins that are not properly folded? They are taken out of the ER and they're degraded by something known as the proteasome, which is in the cytosol, and it will degrade improperly folded proteins because, we don't want those. They are exported from the ER and degraded, like so. So if you think back to our original chart, or the mind map that we drew, after being sent to the ER, some are sent to the Golgi. Some have jobs in the ER, so they will just stay there. They have tags, which tell them to stay in the ER. So trans-Golgi vesicles are coated with specific V-snare proteins that allow them to be targeted to specific compartments. So on the surface of this membrane, we will have T-snare. The T stands for target, the V stands for vesicle. Complementary T-snare will be present at the intended destination, and what happens was there will be a docking of this vesicle and then a membrane fusion that allows the protein to be targeted. And here is a better diagram of what I've just drawn. Now remember, in co-translational transport, we can actually target proteins to lysosomes as well, and the way this works is lysosomal proteins are made in the endoplasmic reticulum, they're transported to the trans-Golgi network, and then targeted to endosomes and phagosomes, which then convert them into lysosomes. So proteins synthesized, the targets to endosomes and lysosomes, this converts them into, endosomes and phagosomes, converts them into lysosomes, there we go. Lysosomal proteins are then tagged with, very important, mannose 6-phosphate. This is very important. This is basically the essence of lysosomal tagging. Okay, this is how you target things into the lysosome by using mannose 6-phosphate. Lysosomal proteins are they're tagged with mannose 6-phosphate in the Golgi, and there is a mannose 6-phosphate receptor in the Golgi which directs protein into the correct transport vesicle. The significance of this in medicine is one example of a disease. This is known as I-cell disease or inclusion cell disease, which is a mutation in the Golgi enzyme, which I still don't know how to pronounce, glucanac phosphotransferase. This enzyme is responsible for the phosphorylation of mannose, and a mutation in this essentially means that your lysosomal proteins are secreted, which leads to the presence of inclusion bodies in the cell, and that is inclusion cell disease, a disease of the phosphorylation of mannose, which affects the lysosomes. And that is pretty much the end of co-translational transport, which means we are going to move on to post-translational transport. If you go back to the flowchart, here is the flowchart, okay? One such example is the mitochondria or the chloroplast. Imagine a big protein, and the C-terminus is the N-terminus. One important thing I'm going to point out is this signal sequence, okay, is present on the N-terminus, always in the mitochondria. What it's going to do is it's going to pass through the membrane, it's got like the receptor, and it's just going to pass through. Important point, this is an irreversible process. The reason I make such a big deal of this is because in the nucleus it is reversible. We're going to explain why. Only proteins with the correct target sequence, basically can be fed through the receptor, and enzymes will cut off the targeting sequence so that gets snipped off. We draw like scissors. Yeah, snipped off. Signal cleavage. And for that reason, the mature protein is shorter than the original protein. One thing I want to point out is that the protein is complexed with something known as HSP70. This is a chaperone. And the role of this is to maintain the unfolded state of the protein, okay? So this is different to the nucleus where proteins will enter folded. Speaking of nucleus, we will move on immediately to the nucleus. Oh, I'm sorry that this pen is running out. Okay, moving on to the nucleus. We've got our little protein. We've got the NLS. This stands for nuclear localization signal. Unlike the mitochondria, the localization signal, this can be anywhere. It doesn't need to be at the N-terminus. It can be here, and it will enter through the nuclear pore complex, and sometimes it may actually be able to leave. Why is this important? So this can go into the pore complex. Terrible drawing. And a protein that enters through the pore complex and stays in the nucleus. This could be an example of a histone protein. But some proteins also, as well as a nuclear localization signal, they may also possess something known as a nuclear export signal. Okay? Now, if you possess a nuclear export signal as well, this means that you can leave the cell through the nuclear pore. Why would you want something to go both in and out? Well, this could be something like a transcription factor or a signaling protein. Signaling protein can go back out. So that's the peculiarity, I guess. Example of nuclear localization signal, PKKRKV. If you are not familiar with the one-letter abbreviation, I would recommend having a look at that. It's proline, lysine, arginine, lysine, valine, which means that this is overall it's basic because of the basic amino acids. The energy for this process is provided by the RAN gradient. What is going on here? What is this? This is the important, also known as the nuclear localization signal. What it does is it's going to go in through this pore complex and it's going to cause a conformational change within this whole pore complex. I'm so sorry about this pen running out, by the way. It's going to interact. Once inside, it's going to interact with this thing, RAN-GTP, which is a GTPase. Okay, that interaction will occur. Then RAN-GTP will export the important out of the nucleus. Lastly, in our series about post-translational transport, one thing we haven't covered yet is the peroxisome. What is going to happen in the peroxisome? Okay, can you actually see with the... Okay, peroxisome. The peroxisome translocation signal is always present at the C-terminus, and it is serine, lysine, leucine. Okay, that is the peroxisome translocation signal. By no means did this video cover everything. This is meant as a sort of introduction, so which is why it's less than 15 minutes long. You might have learned something from it. Thank you for watching.